Another experimental work was conducted by the Institute of Biochemical Physics of the Russian Academy of Sciences. And, well, the article is written by the scientific director of the institute, and she did it in a most careful, diplomatic way. So no direct conclusions, no even mentioning, what is his name, the French guy. No, the homeopathy, Benveniste, right. They worked on the very diluted solutions for 20 years, and they found that the effect is definitely there, but the effect is not uniformed. It can affect the living system in one direction or another direction. For instance, if the cells are under-energized, the diluted solution would add the energy, and vice versa. Anyway, the question is, what carries the memory of water? And the only answer is the structuring memory, structure memory of water. And it was a very convincing set of works showing that the memory of water structures are very short. It is 10 to the minus 13 of a second. So there is no way to assign the effect to structural memory of water. They nevertheless say that, well, this is still one of the possibilities, but you cannot blame them. They are members of the Academy of Sciences. So it is a second work. And they make a huge reference to different works in this field. The third work is, this third article, is what it is Cliff Baxter's work. And in addition to the material in this book, Cliff gave a detailed explanation of the methodology of the work on. Anyway, the methodology is presented properly, and you can repeat it, you know. I mean, not you, but you. So this is the third experimental work, and the fourth experimental work is by a lady from Walter Reed Army Institute of Research, who is a biochemist, and now she is a professor of biochemistry in Virginia. But she brought capable healers who observed a culture of T cells in test tube. And the intent, as she asked them, is to increase calcium concentration in T cells. And indeed, and it is a measurable thing. And she applied the contemporary methodology to determine the concentration of calcium ions inside the cells. This concentration increased by 25%. So this is a typical, most promising work. You bring talented individuals and let them work on the simplest living organisms. And then perhaps we can come close to the relation between the mind or the biofield control system of humans and the biofield control system of a cell, and see the chemical, say, changes. Well, we are talking about how to get closer to measurable things, because otherwise we don't know what to measure. So maybe in this way we will be able to determine or to set up a unit of the interaction. Before we have a unit of interaction, we don't know what we are talking about. When we are talking about space, it is a meter. When we are talking about time, it is a second. When we are talking about electrical parameters, it's a volt, ampere, or whatever, measurable. But if we don't have a unit of anything, we don't know what we are talking about. So the whole idea is to come closer to develop a unit of this biofield, of the nature of this biofield control system. And this is the way, letting the most talented individual work on the simplest organism,